Oops. All right, ladies and gentlemen, welcome to our third lesson of my Unit 0, where we are going to talk about statistical analyses. If this gets to be too long a video, I may end up splitting it in two because, as you can see if you take a look at the guided notes, there's a lot of stuff we're going over, including some sample questions as well. So how do we tend to measure the variation in data? What are some of the common statistics we use? Well, for variation, we tend to use things like mean, median, mode, and then standard deviation as well. Or if you're thinking back to what you may have learned a long time ago in math, mean, median, mode, and range are also some of the things that are used. And my apologies for my sniffles. I think my allergies are acting up. So on the bottom of page 1 of your formula chart, you will see the definitions of mode, median, mean, and range. And if you forget about them on the test, you can look them up there. The only one that I've really seen on your test out of those 4 on the bottom are mean. That's it. Mean, and then standard deviation, and a few other types of statistical analyses that we'll do here. And these are some of the kind of basic things that are used to analyze statistics in biology. However, when you get to a college biology course, you may be introduced to even more complicated statistics, so just keep that in mind. So, what's a mean? Well, you may know what mean is, and I hope you do. It's the average of a set of numbers, and unfortunately, on your formula chart, you get this super crazy complicated formula. So let's take a look at what all the variables are. X bar is the mean, and it represents your sample size, so how many data points you have, how many measurements you took. Xi is a stand-in for each individual data point. So what they're basically saying is that you have X1, your first data point, X2, your second data point, X3, your third data point, all the way to Xn, which would be your last data point. And so that giant E is sigma, it's a Greek letter, and we use it to represent sum or adding all of them up. So what does this end up meaning? That to get your mean, you're going to add up all of your values that you've collected, all of your data points, and divide by the number of data points. That's it. It's the same formula as an average, just for some reason they decided to make it look super complicated, so don't let that fool you, because this is the only reminder of the mean formula you will see on the exam. So we're not going to do a practice problem for mean because hopefully you guys are good with averages. Let me know in class or in tutoring if you want to go over how you calculate a mean again. Now we're going to talk about the standard error of the mean. So when you are taking data and you are gathering results, you're usually doing so from a sample of what's a larger population. It's not practical to take measurements for the entire population. So for example, if we were doing this with height, and if we were taking the height of the average American, we're not going to measure that in all 300 million plus Americans. It's not practical, and honestly, it's a lot of work that we don't necessarily need to do in order to get a good picture of what the actual population data would look like. So you have a sample mean, which is the mean that you calculated from your data. But then we have this theoretical, hypothetical population mean, that if we could calculate the mean of the heights of everybody in the United States, that mean that we calculated there would be our population mean. So how close is our sample mean to our population mean? And that's basically what the standard error of the mean will tell you. So you calculate the standard error of the mean, and then within 2 standard errors of the mean, there is a 95% chance that your population mean is in that within those borders, either -2 SEM or +2 SEM. The sample mean, like I said, is not going to be exactly equal to your population mean, but it's good for letting you see an estimate of what the mean of the population is likely to be. And I just want to point out that when you have smaller sample sizes, that's going to give you a less accurate estimation of your population mean. Obviously, the larger the sample size you can get, the better. So let's take a look at the formula, and this is directly from your formula chart. So X bar, once again, means mean. The S is the standard deviation, and you will hopefully be given that, although a lot of cases you're just given the standard error of the mean and you have to interpret it. And then n is sample size again. And I just want you guys to know, the larger the sample size, the smaller your standard error. So if you have really large standard error and you want to find a way to decrease that so you can be more certain of your results, that your sample mean represents your population mean accurately, you can take more samples. And the larger your sample size, the smaller your standard error. So we're going to do a quick calculation. I do not anticipate you having to calculate the standard error of the mean on the AP test, but just in case, let's check this out. So before we get started, I'm going to go ahead and write my standard error equation up here. So that way, when I'm reading through this, I will have an idea of what I am looking for. So it's standard error of the mean. Already said that. I'm looking for my standard deviation and my n. And those two numbers will help me calculate this. So we've got a group of 50 people and measured their weight. The mean of the group was 65 kilograms. The standard deviation of the data was 5.6. We want to calculate the standard error, so we're going to do some math. And we want to graph the sample mean with error bars of plus or minus 2 SEM. And I have seen multiple free response questions where you did have to do the graphing of error bars, so that's why I'm making sure that we go over this here. So what two pieces of data do we need to calculate our standard error? Well, we need our standard deviation, which they gave us. It's 5.6. We also need our n, or our sample size, so how many measurements did we take? Well, we took 50 measurements because we measured 50 different people. So if we plug that in, our standard error is going to be 5.6 divided by the square root of 50. And I've already pre-done all these calculations just to save time, but you are welcome to pause anytime I say go put this in a calculator and do the math for yourself just to make sure you're entering it correctly in the calculator. I know a lot of you guys are really used to using graphing calculators, and while you can use those on the test, I don't have any to lend to you. So if we're taking this test in person, all I have are scientific calculators, so you'll need to get used to entering data, entering equations, calculations, and that. So when you divide this out, you get 0.79. Then that's your standard deviation. So what you could put is that's your standard error, sorry. Your standard error is 0.79. The next thing you need to do is graph it, though. And you're graphing the sample mean. Well, our sample mean was 65 kilograms. So I am only graphing for one group. So I'm just going to, remember, I got to always label my axes. So we're going to say group 1. If we had more data, we might be graphing another group. And then my y-axis label is what I measured, which is the weight. And you always have to include your units. Otherwise, you risk losing points. So my weight in kilograms. Then I'm going to go ahead and just see if I go up by each each demarcation is like 10 kilograms. That seems to fit the data pretty well because my largest value I'm going to have to graph is 65 plus 2 times the standard error. So I'm going to graph my 65. So let's say that's about here. And once again, my apologies. I'm doing this on a graphic tablet. That makes it really hard to draw straight lines sometimes. So there is my very messy, slightly hairy looking graph, but I'm not done yet because I need to graph error bars. And they need to extend 2 times the standard error above the mean and 2 times the standard error below the mean. So what I can do is I can do 2 calculations. My mean minus 2 times the standard error, and my mean plus 2 times the standard error. If I plug in numbers, that's going to give me 65 minus 2 times 0.79, which when I put it in the calculator, gives me 66.6. Then on the bottom, that second calculation is going to be 65 minus 2, plus 2 times 0.79, and that's going to be Oh, my bad. Sorry, I did this in the opposite order on my scratch paper. 65 minus 2 times the standard error is 63.4. And plus 2 times the standard error is 66.6. Never be afraid if you're dealing with math to go back and look at the numbers and say, wait, does that make sense? Because if I'm trying to subtract something from 65, it shouldn't be a number bigger than 65. So that's just something to keep in mind. So I've got my mean plus 2 SEM, my mean minus 2 SEM, and now I need to add that onto my graph. So this is not going to be very large. I probably should have come up with some data set that has a larger standard error, but oh well. So I'm going to go up to 66, which is probably going to be, I'm going to say around here. It's probably a little lower, but I want it to be visible. And then below that, let's say 63 is around here. And so I'm going to draw a horizontal line where those standard error limits are, and then I'm going to connect them. So this data set that we've gathered, or that I've made up, quite frankly, has a very small range for the standard error. The standard error is very small. So we have some pretty good confidence that our actual population mean of whatever population we're measuring this for should be within that range that I see in the error bars. All right. So now let's talk about rates. In biology, the most common rate we're going to look at is how something changes per unit time, or how something changes over time. If you have a graph where the x-axis, the dependent variable, is time, then a lot of times that rate is going to be the slope of your graph. But be careful, because sometimes it's not just as simple as calculating the slope of the graph. There can be some complicated questions, so make sure you look at what rate the problem is asking you for and what data is present in the graph. Because sometimes you might have to use the data in the graph to figure out numbers that you then calculate the rate from. So here's a fairly simple example. Let's say that this is showing the concentration of a reaction product over time. So we have our units, so I would get points for axis labels on this. And so we want to calculate what is the rate of this chemical reaction between 4 and 8 minutes. So that means that I am not going to be calculating the slope of this entire line. I'm going to be calculating from here at 4 minutes to up here at 8 minutes. And I did say this in class, but when you are doing calculations on the AP test, usually the scoring guide has a range of numbers to allow for the fact that, these numbers aren't directly on a line, so you're going to be estimating their values, which means that you could be off by a little bit. Just try and get as close to accurate numbers as you can. So, like, for example, if I said that, that value at 8 minutes was 50, that would be clearly wrong. It is something under 40. All right, so I said that a lot of times what the rate calculation is going to be is it's going to be the slope of the line. So slope, and hopefully you had this in algebra, is your change in your y over your change in your x. The way I always learned it and remember it is rise over run. How high does that line rise over how long it's running? Okay, so I need to get my value for 8 minutes. At 8 minutes, my concentration of the product looks to be about, let's say, 39. And I'm just going to put the units everywhere because I don't want your chemistry teachers coming to me and saying, you got my kids out of the habit of writing units and everything. Then I'm going to subtract my value at 4 minutes, which looks to be around, let's say, 16. And all of that is going to need to be divided by my difference in my x value, which is 8 minutes minus 4 minutes. Then, when I calculate that out, When I calculate that out, what I get is I get 5.8. And as for units, because when you are giving an answer, always include the units, it's going to be milligrams per milliliter, and if I want to make this really messy looking, per minute. And there's my answer. Now, ratios and percentages are also things that you're going to need to be able to calculate. A lot of times this is going to be with genetics, but it's not always genetic. So a ratio is basically showing the relation between two amounts. It's usually expressed as a number, then a colon, then another number. So an example of a ratio would be this right here. So if we had a class where we had twice as many males as females, we would say the ratio of males to females in this class is 2 to 1. So for every 2 males, I have 1 female. For every 1 female, there are 2 males. This is commonly used to show when you're doing a genetic cross and you're predicting approximately what percentage or what fraction of the offspring will have a particular trait or have a particular genotype. That's going to be where we commonly use ratios. So we have a sample question. We've got a plant that's heterozygous for flower color, and since we haven't gone over genetics yet, and I don't know how much you remember, I went ahead and I told you what the genotypes are. So it's a big R, little r, is crossed with a homozygous plant, recessive plant, which is little r, little r. What is the predicted ratio of offspring that will result from this cross? And since I haven't told you anything about what the flowers will look like, I'm just looking for a ratio for the genotypes, the genes that these organisms will inherit. So I use my old friend the Punnett square, but later on we'll talk about other ways you can do genetics without Punnett squares if you don't like them very much, but I put the alleles from one parent on one side, the alleles from the other parent on the other side, and I get this. And if you don't remember how to do Punnett squares, that's fine. We will be going over it later. So if I count up, I have 2 different genotypes that come out of this. I have 2 out of 4 are going to be big R, little r, or heterozygous, and another 2 out of 4, the remaining 2 out of 4, are going to be homozygous recessive and be little r, little r. What this means is that for every 2 big R, I have 2 little r, little r. And while I think that's technically acceptable on the AP test, in general, we like to simplify things. So for example, 2/4 can be simplified to the fraction 1/2. Same here, 2/4 can be simplified to 1/2, which would give us a 1-to-1 ratio. So for every homozygous recessive plant we had, we would get 1 heterozygous plant, and that's my ratio. Not too shabby. All right, percentages. It's a number or a ratio that you express as a fraction of 100. That's literally what percent means. Per like each, and cent like centavo, like a cent, like the number of pennies that go into a dollar. All of that is based on the fact that cent means 100. So it's per 100. It has no units, so you don't have to remember to put a unit on it, but you should remember to always put the percent sign just so it's clear what you're providing. And it is always a part of something over a whole something. So for example, we're going to do another example, but let's say I was looking at that class where for every 2 people, 2 males, I had 1 female in the class. That means 2/3 of the class is male and 1/3 of the class is female. If I wanted to calculate the percentage of the class that was female, I could do the part 1 out of every 3 is going to be female. So that's the part over the whole. And then I can multiply it by 100%, which is going to give me 33.3%. Now let's do a sample problem. So another genetics problem. And I'm going to go over the genetics part super quickly, and we'll go over this again later in the year. So in sheep eye color is controlled by a single gene with 2 alleles. When a homozygous brown-eyed sheep is crossed with a homozygous green-eyed sheep, blue-eyed offspring are produced. If the blue-eyed sheep are mated with each other, what percent of their offspring will most likely have brown eyes. So let's say we have brown and we have green-eyed sheep. So you've got one that's got 2 genes for brown and another one that's got 2 genes for green. Well, if those two mate, all of their offspring are going to have one brown gene and one green gene, and that's going to make them blue-eyed. So if we have 2 blue offspring mated with each other, that's going to be BG by BG. If I do that, I want to know what percentage will be this, brown. So once again, I can do a Punnett square. And I'm looking for all of my BB, my big B, big Bs. That's 1 out of 4. So for every 4 offspring, one of those should have brown eyes. If I try and turn that into a percentage, well, it's my part, which is 1, divided by my whole, which is 4, times 100%. Stick that in the calculator, and it would give you 25%, which means your answer would be B. So let's go more complicated because those were not super complex. And if they were for you, don't worry, I have tutoring. So let's talk about normal distribution and standard deviation. So what is standard deviation? Standard deviation calculates how spread out the data is. And this is compared to your mean, your average. So if your data points tend to be clustered pretty far from your mean, it means your data is more spread out and your standard deviation is going to be higher. It means you have more variability. You are not going to have to calculate standard deviation on the AP test, but you need to understand what the statistic means, and you need to be familiar enough with the formula to be able to draw some conclusions. So let's look at these 2 sets of data. They both have the same mean. For both of them, their mean is 5. But they're not identical sets of data. If you'll look, we've got a lot more spread on histogram 1. In fact, just put it up there. Histogram 2 has less spread, less deviation. And the standard deviations for these were calculated. You can see the one with the more spread, the one with the wider set of data, has the larger standard deviation. The one with less spread, the narrower range of data, has the smaller standard deviation. So let's take a look at the formula. So S stands for standard deviation, which we saw in the standard error formula. X sub I stands for each individual data point. So you're going to have to do this calculation for every single data point. Then X bar is your mean. N is your sample size. And our good old sigma there that looks like a giant E is our sum. And just wanted to point out that if you look at this, there's a relationship between the sample size and the standard deviation. If you increase the sample size, that's going to make the number on the bottom of that fraction bigger, which will end up making that entire number lower. So when your sample size goes up, your standard deviation goes down. And this is a reflection of the fact that with a larger sample size, you can be more confident that your sample results reflect the population results. So we're going to do basically a kind of test calculation, but I want to point something out. So on the AP test, you won't have to do this calculation. Hopefully, if you ever have to do anything like it, they'll give you a table like this, which lets you do each step of the problem bit by bit. But let's go ahead and let's fill this out, and then we'll do the final calculation. So I have 3 people's weight, and I have gathered their data. And so now I want to know the standard deviation of this sample. Well, first thing I need to do is I need to figure out what is my N. How many samples do I have? I got 3. Then I need to calculate my mean, which means I need to add all of these up and then divide by my N, or the number of samples, which is 3. And when I calculate that, I should get 65. The next thing I'm going to do is for each individual data point, I need to take that data point and subtract the mean from it. So basically, it's like I put minus 65 after each of these data points, and that will give me these numbers: negative 13, 13, and 0. My next step is to take the value that I got in the previous table, and I'm going to take that value and I'm going to square it. So I'm just going to take this number right here, square it, and that's going to go there. So negative 13 times negative 13 is 169. Same here. I'm going to square it, and 13 squared is also 169. And then hopefully you know that 0 squared is 0. I'm going to add all of that up, and that gives me 338. Now, if you remember what the standard deviation formula looks like, we have so far only calculated everything that's on top of this fraction, which was a lot of stuff, but now we need to plug everything back into this equation. So my standard deviation is the square root of the sum of all of these xi's minus x bar squared over n minus 1. When I substitute stuff in, well, all the stuff on the top of that fraction equals 338. And on the bottom of that fraction, I know my n is 3, so n minus 1 is 2. And then I'm going to need to take the square root of all of it. And when I do that, I'm going to find out that my standard deviation is 13. So I may have a fairly wide spread. It's kind of harder to say since I only have 3 samples, but that suffices for a sample question. Now, when we plot a lot of data in nature from biological experiments, a lot of it tends to fit this pattern, where you'll have most of the data will be centered close to the mean, and then you'll have less and less of the data farther from the mean. And it'll form this nice, pretty, symmetrical distribution that's distributed or spread around the mean. And then what this diagram is telling you is this symbol stands for, sorry guys, helps if I actually press with the right end of the pen. This stands for 1 standard deviation, so plus 1 standard deviation above the mean, and this stands for 1 standard deviation below the mean. So that means within 1 standard deviation, whatever number you calculate of the mean, you should find 68.2% of your data points. And this is when you have a normal distribution. On the other hand, when you have 2 standard deviations worth, that's going to be 64.2 plus, let me see how to draw this one here. And sorry, those look like weird bats. So that's going to be 68.2 plus, sorry, don't know why I made there a decimal, plus 27.2. Sorry, mental math. So it's basically going to be, it looks like around 95%. So 95% of your data should be within 2 standard deviations of your mean. And though the spread of this curve is dependent on your standard deviation, or is going to determine your standard deviation and let you draw conclusions about the variability. So the wider the curve, the larger your standard deviation, the greater range of values you have aside from the mean. If you had a normal distribution that was very narrow, that would mean that most of your data points were falling fairly close to your mean. All right, now let's talk confidence intervals and the error bars that go with them. So what is a confidence interval? It is, just like with standard error of the mean, trying to figure out how accurate is your sample mean compared to the mean of the entire population. So if you're taking a mean from repeated random samples, your confidence interval means that 95% of those samples you're taking would, or within 95%, within that range, 95% of your samples would include the population mean. It's often represented by error bars on the graph. This can be done on line graphs and bar graphs, and you won't have to calculate this on the AP test, so I'm not even going to talk about the formula. Instead, we're going to talk about the error bars because this is what tends to commonly show up. So if you're graphing, you can either graph error bars with 95% confidence intervals or plus or minus 2 standard error of the means, depending on what data you're given. And usually on an AP FRQ question, they will give you that value that they want you to graph. And what you're doing with this is you're trying to see, are these results statistically significantly different? You're looking for a statistically significant difference between your two groups of data. If your error bars don't overlap, so what that means is if there's no point at which your error bars like meet up or overlap each other, then you have a statistically significant difference. So in this experiment 1, the control group and the treated group are statistically, they've got a significant difference, meaning that these results, these differences you see, are not going to be likely to be due to chance because we're fairly confident that the range of that population mean for the control group isn't going to have the same value as the population mean for the treated group. However, let's say you've got slightly different data. Your error bars do overlap, meaning that when you look at the bottom of the one on the left and the top of the one on the right, they overlap. Since those error bars do overlap, your population mean, if it's in this range, they would have very similar population means, and you wouldn't be able to say that there's any statistically significant difference between the two groups. So the most common thing you'll be asked about error bars is there a statistically significant difference, or what do these error bars mean? And this phrase right here, statistically significant difference, is super important. So if you're asked about error bars or what error bars mean, that is very important to include in your answer. And last, but most certainly not least, your chi-square analysis for goodness of fit. It is not chi-squared, it's chi-squared. And this has to do with null and alternate hypotheses, which we covered in the first lesson on experimental design. So if I start talking about null hypothesis or alternate hypothesis, and you've got no clue what I'm talking about, go review that video, please. So what is this? What is this? Basically, you make predictions about your data. So if the results were completely due to chance and there was no relationship between your two variables, what data would you expect? And that's this guy right here, your expected results. And then you're going to check the actually observed, measured results against that to see, well, are these differences due to chance? Or, is it statistically likely that, oh, hey, there's something going on here other than my variables? So a chi-squared test is usually going to try to reject a null hypothesis, or the way we say it is you fail to reject a null hypothesis. You don't say you accept another one. So you're basically, you're building up evidence against your null hypothesis that all your differences in your data are just due to chance. There are several topics that use this. Any place where you've got kind of these expected values that you can compare to will let you do a chi-squared problem, but most typically it'll show up in genetics problems. You might see it in a behavior lab, which we'll talk about later. If we get to go back to school physically, all of us, we might do a behavior lab, a mitosis lab, and Hardy-Weinberg equilibrium problems. And if you don't know what that is, that's fine. We'll go over it with evolution. So this is the formula for chi-square. It's fairly similar in complexity to the standard deviation one. So this is your calculated chi-square value. This is your observed value. So that's the data that you collect. This is your expected value, which is what you would get if the results were 100% due to chance. And this is your sum. So we're going to be adding a bunch of stuff up. Before you can get into that, you need to have knowledge of two different things. One is the degrees of freedom. And I am going to put a link to a website that has a really great explanation of degrees of freedom in the description. It's the best explanation I've ever read. I honestly would not have been able to explain degrees of freedom before I read that. Once I read it, I was like, oh, that makes sense. So let's say we're doing an experiment with peas. And the peas have two different colors. They can be yellow or they can be green. Well, my degrees of freedom is going to be my number of possible outcomes. So how many possible options do I have? Minus 1. So in this case with these peas, my degrees of freedom is going to be n minus 1. It's going to be 2 because I have two possibilities. You can be green or you can be yellow, minus 1. So I have 1 degree of freedom. Then I'm going to need to use a table that's actually found on your formula chart. It gives you what are called critical values. These are critical because you're going to compare your chi-squared value to them, and depending on which number is bigger, you're either going to reject your null hypothesis or not reject it. And I know this probably sounds very confusing. We will do multiple practice problems, and especially once we get past genetics, this should be a little easier to understand, or at least a little easier to say. And so what you're going to do is, based on what your what your degrees of freedom is, you're going to use that number to select what's called a critical value. And unless you're told otherwise in a question, we always use this top row for what is called a p-value of 0.05. And I'll explain p-value on the next slide. And so we had, in the previous slide, we had 1 degree of freedom. What that means is that our critical value for a p of 0.05 is going to be 3.84. So if your chi-squared is greater than your critical value, then you're going to reject your null hypothesis. If your chi-squared value is less than your critical value, you fail to reject. Never say you accept it. Why? Because you can't fully prove your null hypothesis. You can only provide evidence for or against it. It's a statistics thing, but you can lose points, perhaps, depending on how strict they are the year of the AP reading, if you say accept null hypothesis. So be careful about that. So what is a p-value? A p-value is basically a probability, and it's very commonly used in science to determine how significant your results are. You'll see if you read research papers, if you're looking at data, a lot of times the main p-value that you'll see is 0.05. And what that means is there's a 95% chance that the difference you see between your expected and your observed values is due to something other than chance. And it's 95% confident, so that's pretty confident. If your p-value is less than 0.05, that's really strong evidence against your null hypothesis, and so that will give you more fuel to reject that null hypothesis. If your p-value is greater than 0.05, that's generally weak evidence against your null hypothesis. It is not statistically significant enough for you to say, no, I'm not going to reject the null hypothesis. If you want to be able to reject it, you're going to have to do more experiments. So the p-value is basically the probability of obtaining these observed results, assuming that the null hypothesis is correct. So smaller p-value means stronger evidence against the null hypothesis. So let's look at a sample question here. So first, I'm going to give you the null hypothesis because the null hypotheses are a little weird for genetics. But basically, your null hypothesis is that offspring will have the expected ratio of colors based on our Punnett square. So to go through all of this and simplify it, we've got homozygous green peas, we've got homozygous yellow peas. So we've got 2 different possible colors. That means we can be yellow or we can be green. All right, that tells us, well, we've got an n of 2. Our degrees of freedom is 2 minus 1, so that's going to be 1. All right? Then we're saying that all of the offspring were phenotypically yellow. So if we crossed homozygous green and homozygous yellow and all the offspring were yellow, that tells us yellow is dominant. So this was our parent cross, and they're going to produce all heterozygous babies. Then what the F2 generation means is we're going to cross those heterozygous babies with some other heterozygous babies, and we want to figure out what's the ratio of offspring you expect. And if you want, we can go over in tutoring or office hours how to do this in more detail. I'm just going to tell you it's going to be expected a 3-to-1 ratio. For some reason, I always write ratio on accidentally. It's a 3-to-1 expected ratio of yellow to green. So then we're told that we got 20 offspring out of this. So that means our observed is 20 total. And we want to predict how many will be green and how many will be yellow. So if we had 20 total offspring, for every 1 green pea, we should get 3 yellow peas. What that means is I should have 3/4 yellow and 1/4 green. So to get my expected number, I basically just take that ratio, so 3/4 times my total. And then for the green, it's 1/4 times my total. And when I do that math, I'm going to get 15 and 5 as my expected values for yellow and green. Now they give us some data. They said if the offspring consisted of 8 yellow peas and 12 green peas, would this be statistically significant from your prediction? Or statistically different. All right, so our observed values are 8 and 12. Keep in mind, this total adds up to 20 in both the expected and the observed columns. Then we're just going to fill this in using this table. So our observed minus our expected at the top is 7. For green, is -7. For green is +7. In the next column, I'm going to square these terms. And that's going to give me 49 and 49. Then I'm going to take this value of 49 and I'm going to divide it by what I expected. So it's going to be for the yellow, 49 divided by 15, which is going to give me 3.3. For green, that's going to be 49 divided by 5, which is going to give me 9.8. Now, my chi-squared value is what happens when I add those together. So I'm going to add them up, and when I do that, it's going to give me 13.1. So for this, I need to know what my critical value was. And let's go back to the previous slides and get it. For 1 degree of freedom with a p-value of 0.05, my critical value is 3.84. So now I need to see what's bigger, my chi-squared value or my critical value. Well, when I look at it, my chi-squared value is 13.1. My critical value is 3.84. Which one's bigger? My chi-squared value. So my chi-squared value is greater than my critical value, and you should have seen that when we did that, we would get, we would go back and we would reject our null hypothesis. What that means is these are not being inherited in the manner we expect. We would expect to find 3/4 or somewhere close to 3/4 of these offspring being yellow, but that number 8 out of 20 is too far off. Something else must be playing a role. There must be some other factor influencing the distribution of these babies. So does the data fit the predicted phenotypic ratio? No, it does not. So that is the end of our lesson for now. We will be using these statistics throughout the year, so anytime you are confused, please come back, watch bits of this video. There will be in the description, there's going to be a link to that website that I said has the best explanation of degrees of freedom I've ever seen. I'll see if I can find some good chi-square and standard deviation websites to throw in there too, just so you can go and look some of this stuff up if you're still having trouble understanding it. And then fill in the blank notes will be in Google Classroom shortly. All right, I'll see you next time when we start Unit 1.